Hello, welcome to Psychiatry Grand Rounds. It's my pleasure to introduce Dr. Ashley Acheson, a professor in our Department of Psychiatry at UAMS and director of the Neurodevelopmental Risk and Resilience Program. Ashley grew up next to a circus in Baraboo, Wisconsin. Both Baraboo and the nearby state park Devil's Lake are mentioned in Johnny Cash's hit cover song, I've Been Everywhere. Ashley left Wisconsin to pursue his Ph.D. in behavioral neuroscience at the University of Buffalo SUNY, where he studied rodent models of decision-making and impulsivity. He then pursued two postdocs, one at the University of Chicago to study decision-making and impulsivity in humans, and a second postdoc to translate this into neuroimaging at the University of Texas Health Science Center at San Antonio. He joined the faculty at University of Texas in 2008, and then joined UAMS in 2016, and was then promoted to professor last year. Dr. Acheson focuses on neurobiological risk factors for substance use disorders, and lives with his wife, young son and daughter, and their dog Lulu in a quiet neighborhood in West Little Rock. Today, Ashley will be speaking to us about his work in the Healthy Brain and Child Development, or HBCD study. So with that, Ashley, I'll hand it off to you.
Thanks, Andy. I think that was there was also a newer version of that where I mentioned getting hit by a car in Buffalo, and also somebody on the subway trying to let me on fire in Chicago. So
goodness, that was not, those edits weren't in my version.
that's probably good. Fair enough too. That's probably for the best. Okay. So hold on here for a second. Okay. So disclosures. Okay. So this is kind of a different sort of talk today. Normally I'm used to talking about a lot of data. Unfortunately, today I'm going to be talking a lot about kind of background and procedures, stuff like that. But the main things I want people to take away from this is, so what's the Healthy Brain Child Development, or HBCD study? Why is it being done? What have we already done for it? What's next for HBCD? And what opportunities does HBCD give us? So let's go ahead and get started then. So what's the HBCD study? This is the largest ever study of childhood brain, cognitive, and emotional development ever conducted. We have started, as you see, we're starting to launch this study now. There's 25 participant sites. It's funded by 12 NIH institutes. It's going to follow 7,500 children from the prenatal period to at least the ages 9 to 10. It's trying to look at both, so it's looking at how prenatal substance exposure and other environmental factors will affect developmental trajectories. And the data is going to be shared through annual releases. We're expecting the first data release will probably be about 2026 or so. Okay, so this is just showing you who is funding HBCD. And if you notice there, when we say, let me just go back one slide for a second, or two slides here, sorry. So the reason why we spell this weird is because of HEAL is one of our major funders for this. Okay, so it's Healthy Brain Development, Child Brain and Child Development Study. HEAL, NIDA are major funders, and there's also these other institutes that are funding it as well. So why is this being done? So partially it's in response to the replication crisis. It's also another major goal is just to try and comprehensively study normative childhood brain development. And also to better understand impacts of prenatal exposure to opiates and other drugs. So let's start with the replication crisis. So basic problem is many published findings can't be replicated because of things like underpowered samples leading to false positive findings. So, basically it's like the smaller sample you have, the more likely that the results you get, like significant results you get, may actually be due to chance. That if you had a larger sample, that they might be, you'd see more a negative effect. And I don't know how this happens as commonly anymore, but when I was coming up through training, stuff like that, a lot of times in the labs I worked in, once we got significant effects, the study was stopped and we tried to publish them. Okay, we didn't keep on collecting more data, just collect more data. Once we got a significant effect, that a solid significant effect, we would oftentimes stop the study. There's also something called p-hacking, and there's different ways of doing this, but basically it's trying to look at your data in a lot of different ways till you get a significant result. Okay, so that could be things like just, having a big data set and you're just looking for correlations and just kind of like, well, that's whatever correlation you find, try to make a paper out of that. It could be things like trying to basically analyze your data in a lot of different ways till the effect you're trying to find is significant. There's also a major bias for publishing positive results. So if you have a study and you get negative findings, it's a really hard time to find a home for that study. So what can happen is that let's say 3 different groups do an experiment, 2 of them find a negative result, the other one finds a positive result, the positive results get published, the 2 negative results don't get published. Okay, so that can bias the field in thinking. And then if you looked at those all together, that may be more likely that effect may be more of a chance thing too, but that's not what gets published in literature. And there's also a major tendency that we want to overgeneralize our findings. So when I do a study here in my titles, I tend to talk about the population of interest. I don't say that it's a sample recruited from the central Arkansas using convenient sampling. I talk about the study, the sample I'm trying, like the population I'm trying to study, not my specific sample. And it's not always, sometimes the findings we get in samples in certain regions don't necessarily generalize to other areas. So in response to the replication crisis, so basically the problem is like when you have all these findings out there that you can't replicate, then we can't build on it. I mean, science is supposed to take existing literature, use that for a rationale for new experiments, but when the foundation's faulty, that doesn't work. So in response, there's push to things like robustly powered samples and analysis, so much bigger sample sizes, and also representative sampling. So trying to make it, make sure that your sample looks like the general population that you're trying to target. There's also NIH is like funders like NIH are really pushing towards increased attention to things like scientific and methodological rigor. So if your primary rationale for doing a study is just that you had found some interesting pilot data, it's going to really be hard to get that through study sections these days. There's also push to make data publicly available. So like when you publish a study, so now even like for my other grants, not the HBCD, when I publish findings, now I have to make my data publicly available so somebody else can look at that and analyze my data in a different way, or it's all basically trying to increase transparency. So again, HBCD is done partially in response to replication crisis. Another goal is trying to comprehensively study normative child brain development. And what do I mean by this? So there's some basic questions we're trying to answer. So one is, what are typical neurodevelopmental trajectories of brain development from birth through childhood? And how do biological and environmental exposures affect these developmental trajectories? How do genetic influences interact with those environmental factors to affect those developmental outcomes? And are there key developmental windows for exposure? Okay, so so it seems like things like adverse childhood experiences, that there's certain time periods when those are more impactful. So it seems like, things that happen earlier on seem like they may be having a bigger impact than things that may be happening later. And related to that, what's the impact of the parent-child interactions on the later health outcomes? So what we know, I mean, there's a lot of data like this. This is just showing, this very busy slide is showing different aspects of brain development over the first 2 years of life. And you see, especially things like cortical gray matter volume, it's really rapidly increasing in the first 2 years. So we know this, but what we're trying to understand is how different things can modify that, like how different things in the environment, how different genetic factors can modify that. And that's what, so when I start talking more about the HBCD protocol, we're trying to throw the kitchen sink at these families and trying to really understand a lot of different factors so that we can go in and look at how these multiple things can impact these development outcomes. Sorry. Okay. So again, it's being done in response to replication crisis and to comprehensively study normative child brain development. Also, we're trying to better understand impacts of prenatal exposure to opiates and other drugs. So about every 15 minutes, there's an infant born with neonatal absence syndrome in the United States. Between 2004 and 2014, the incidence of that increased 433%. It's now present in almost 1 out of 100 hospital births. 2017, 7.3% of pregnant women used drugs of abuse. So this is where this is the thing that's where things kind of, so we know that having those exposures is linked to greater risk for things like substance use disorders, ADHD, cognitive disorders, anxiety, and other conditions too. But it's hard to say that's just due to drugs. So a lot of my work outside of HBCD is looking at effects of family histories of alcohol and other drug use disorders. And with those groups, even if there's no maternal abuse of drugs, you see a lot of the same increased risk factors for all these things. So just having the family history, family history and things that tend to go along with environmental things that tend to go along with family history, it seems to be linked to all these things that have been directly linked to the abuse of opiates and other drugs during pregnancy. So what we're trying to do is really trying to better understand what is the actual impact of opiates on these outcomes. So, so the things that HBCD is basically trying to answer. So what are the norms? So it's basically trying to wear 2 hats here. So one is, what are normal neurodevelopmental trajectories of children and how these trajectories are influenced by genetic and environmental factors. And then at the same time, it's also trying to be a study of trying to really drill down and what's the effects of prenatal opiate exposure, exposures of opiates and other drugs. So it's kind of both trying to be a normative developmental study and also this one that's really kind of drilled down in the effects of prenatal opiate exposure. And a lot of that, again, is kind of the way that our funding is set up. So, like the HEAL Institute, which is focused on opioid use disorders, is a major funding contributor. So therefore, that's really kind of a big reason why we have such an emphasis on prenatal exposure to opiates and other drugs. Okay, so generally, so look at these different dimensions. There's looking at I don't know if there's questions. Oops. Okay.
I know those are.
Okay, good. We're good. Yeah. Okay. So look at these different dimensions. So psychosocial factors, maternal influences, adverse early exposures, cognitive factors. We're also trying to look at protective factors as well. Biological factors, genetic influences, how those interact to look at for these outcomes. Again, things like risk or protection from behavioral disorders, substance use, cognitive dysfunction, other diseases and illness. Okay, so what have we already done for HBCD? And actually, it's a lot. We've already actually been working on a study for a few years now. So there was a phase 1, which is already completed now. That went from 2019 to 2021. We were one of 35 sites that were funded for this. And for that, we focused on doing pilot data collection to kind of support what's going to be the future full-scale HBCD study. And we also did a lot of work on planning for the phase 2 study, so that the big launch new study. And what that consisted of was lots and lots of work group meetings. So we were really the point of the spear on use of Zoom. So everyone's complaining about transitioning to Zoom in early 2000. We'd already been doing that for a year and we're already sick of Zoom by that point. So we have lots of Zoom experience to this project. Phase 2 is ongoing. This is the big project. Okay, and that's going to go, that started in 2021, expected to go for maybe about 2035. Depending on how well this goes, I mean, it's, like we have this cohort we're trying to follow. The kids are 9 to 10 years old. So if this is all going well, maybe we're going to try and keep following it beyond the ages 9 to 10. I mean, we'll see. So it could potentially be going longer than that. 25 sites were funded. And these are sites across the United States. So the red are data collection sites. The administrative core is in green. That's at San Diego and Harvard. The data coordinating center is split between San Diego, WashU, University of Minnesota, and University of Minnesota. So, sorry. there, and here's our team. So HBCD Arkansas, it's between, this is jointly between ACRI, Arkansas Children's Research Institute, and UAMS. And you can see here, this is a list of our investigative team. The PIs are me, Lorraine McKelvey, and Jiawei Wu. And that here's our co-investors. You can see we have a lot of highlighted the people from PRI. That's overall the most representation of any department is from PRI. Okay, so what have we done? So since we've been funded for phase 2, starting in the fall of 2021, we have been doing a lot of work on protocol development and trying to finalize the protocol. Again, working in the work groups, there's over 24 work groups and other committees. And for right now, we're focused on trying to develop the part of the protocol that's going to go from the prenatal period to age 4 for the kids. And the follow-up, the 4 to 10 is going to be developed later. So just an example for what we're doing for the study. So what I do for work groups, I'm on the steering committee for the first year. I'm going to be rotating off this November or December, rotating off of that one. Work group chairs committee, study design, social environmental determinants, diversity, equity, and inclusion, and also a measures review subcommittee from the diversity, equity, and inclusion work group. So that's just my work group participation. A lot of all my the other PIs and co-Is are also doing a lot of these groups as well. Okay, so what are we going to look at for HBCD? What are our measures? So these are the different major domains. So we're looking at EEG to look at brain activity. So when you to look at brain development with children, so with an MRI, they have to be very still in a scanner. And you imagine that's kind of a problem for a very young child. So what we actually do for the very young children, from infants until they're probably about 2 to 3 years old, is we actually put them in the scanner while they're asleep. So they get we let them fall asleep. We gently carry them and put them in position of the scanner and do our scan and try to get as much of our scanning as we can done before they wake up. And then, so that's what we use EEG to measure activity while they're awake and responding to things. So they can have them doing tasks and stuff like that, looking at different things while they're awake and able to move with the EEG. We're looking at doing biospecimen collections from both the moms and the children to look at substance use and also look at genetic factors, epigenetic factors, nutrition levels, toxin exposures. COVID-19 was going to, this is a bit older slide. So COVID-19 is a bit harder to really try to tease apart when the recent exposure is. But yeah, that's another discussion. Substance use and other risk factors, protective factors and other exposures. We're doing a lot of behavioral assessments, so parent-child interaction assessments, observational stuff with the children, and neurocognitive assessments of the children as they age. We're also doing a lot of work with wearable technology. So the kids are getting at biosensors, look at things like heart rate variability, sleeping, and movement and other activity. So here is our protocol from prenatal to age 4. Again, the older, the one for it's going to follow kids from ages 4 to 10 is going to be developed later on. We're really trying to get a cluster of assessments up front, because that's when, like, if you remember before when that slide I showed you about the brain development, there's some really rapid developmental changes happen early on. After that point, so like in the first year of life, from 0 to, so we're getting, so we do a prenatal visit we do a prenatal visit during pregnancy. We get biospecimen collection, and we do collect different interview and questionnaire assessments from the mom. Then from at 0 to 1 month old, the kids come in, they get for their first, the kids will come in. We're going to do both the biospecimen collections and the assessments on the mom again. We're doing the wearable technology, and they also get their first MRI at this time. Visit 3, they get a lot of those same measures again, with the addition of EEG. Visit 5, EEG again, MRI again. We are dropping the wearable sensors for that one, and same for visit 7. We also do some virtual visits between 9 and 15 months, 18 to 30, and 36 to 60 months. So there's what I was saying before, though, there's a cluster of visits up front. So in the first, there's 3 visits within the first 15 months of life for the kids. And unfortunately, these are looking to be very long sessions so far. So like these are several hours, like with all the assessments we're getting, and also to try and get a kid ready for a scan. Altogether, we're looking at like several hours per each one of these visits. So I think the protocol likely is going to be right now we're doing our piloting. Most likely we're going to not cut down on these different domains, but within these domains, we're probably going to have to make them a bit shorter because it's a very long protocol right now. Enrollment targets, let's talk about that for a minute. So across the whole cohort, we're trying to get 7,500 mother-child dyads enrolled and retained for the study. 25% will have prenatal substance exposure above study thresholds. I'm going to be talking about those in just a minute, what those thresholds are. 75% with no or below threshold prenatal substance use. So broadly across if you remember that map I showed you with the different sites, part of the reason why those sites were selected is try to collectively capture the kind of be representative of the United States population, both in terms of like, racial, ethnic things, and also like income levels, rural versus urban, and different things like that. So trying to get a sample that looks pretty similar to US population this time. So our local enrollment targets are going to be based on what our catchment area is. So we identify counties that we're going to be recruiting from, and then they look at like what are the makeup of those counties, and they give us targets for trying to recruit from those counties. So that's to start with. But what's going to likely happen is we're trying. So we basically have an enrollment monitor across the whole site, and we're looking at how we're filling these different demographics and how we're and if certain sites are really having a hard time with recruiting a certain population, then what they're going to look for other sites that may be doing better at those populations. So let's say that we're really good at recruiting, say, moms with prenatal opiate exposure. We could get asked to try to recruit more of those if other sites are lagging, to try and build up the target we need for the overall study. Okay, so the prenatal substance exposure criteria, I just want to talk through what these look like. So about half of the 25, about half of that, they're targeting for opiate exposure during pregnancy. And these are the criteria that they're giving us for opiate exposure. So somebody who's in treatment for an opiate use disorder, use of prescribed opiates for a period of 2 weeks or longer during pregnancy, positive toxicology in the medical record, if the kid has, when the child is born, if they have neonatal absence syndrome, or if they test positive on any of the biospecimen collections we're getting so that we collect biospecimens from the mom during pregnancy, and at the neonatal visit at this point. And we may be increasing that a bit, which I'm going to talk about later, but for right now, that's what we're planning on. For alcohol, it's a little bit more lower bar to meet. And so just before I go into alcohol though, for someone with who has opiate use that doesn't meet these thresholds, that means they would go into the 75% non-exposed group. Okay, so if somebody, let's say that they use opiates, that they had a prescription of opiates for a week during pregnancy, they'd be considered non-opiate exposed by these current criteria. So for alcohol, though, it's a bit lower threshold, though. So that for alcohol, they only need to have 6 or more standard drinks per week for 2 or more weeks during pregnancy. So you imagine somebody who, say, didn't know they were pregnant and went to a party or went out to a bar a couple of times, they could easily meet this threshold. 3 or more drinks per occasion on 2 or more occasions during pregnancy. Again, not hard to do that. Or if they have a newborn diagnosed with fetal alcohol syndrome. And we also say a positive research collected biospecimen, but reality, that's really hard to do. So a lot of the biospecimens out there that can pick up alcohol use, you can catch alcohol use with a breathalyzer, but beyond that, to actually like detect like evidence of alcohol use, it's really hard. People generally have to be drinking a lot to be showing up for these different biomarkers. Cannabis, again, not very high bar for this. Weekly use or greater than or greater should be four greater. Weekly use or greater for 1 month or more during pregnancy. Okay, so 4 times during pregnancy, they would count as cannabis exposed. Positive toxicology in the medical record or a positive research collected biospecimen again. Tobacco is also fairly light, so weekly use or greater for 1 month or more during pregnancy, if it's noted in their medical record, or if they have a positive research collected biospecimen. So again, for opioid, we have a fairly kind of a somewhat higher bar to, but they don't have to be an opiate use disorder, but somewhat higher bar for opiate use. But for the other ones to kind of substance exposed, it's fairly light. Okay, so what have we done again for the study? We've been working on protocol development. For this study, we have to get SMART IRB approval. We're working through a SMART IRB. And so for people who aren't familiar with that, I'm just going to talk briefly about that. SMART means streamlined, multi-site, accelerated resource for trials. And so this is a way for multi-site, it's used for multi-site studies. Here, what we have is the HBCD administrative core gave all the sites templates, IRB templates that where we have to make local context edits. Then we send those to our local IRB for review, and then once they're okay with that, they cede regulatory oversight to the central IRB, and then away we go. And so when we make modifications, we have to go through the central IRB to get for approval for those. Site certification. So we have the protocol, staff all had to be trained on, and Al can talk about that. There's a lot of extensive training going on to run this project. Even just the things like enrolling subjects or setting them up, and consenting them, lots and lots of, it's very involved to do that. It's all done through things like REDCap and different databases. So the staff needed to be trained on the different data collection procedures, and then need to be certified. So what they have is the San Diego staff basically monitor and improve the local staff while they're performing different study components, which is fairly stressful for people having to go through that. We are currently piloting the first 3 visits: the prenatal visit, the neonatal visit, and the 9 to 15 month visit. And then we need to get successful completion of 5 to 10 participants across the different study components. Okay, and then what's next? Okay, so based on the pilot data that we get back, we're going to finalize the visits 1 through 3. Once that protocol is finalized, we're going to start a regular enrollment. Okay, and again, across the whole cohort, we're enrolling 7,500 mother-child pairs. We will have a 3-year enrollment period to do this. We're targeting 300 enrolled locally. Again, 3-year enrollment period. And then while this is going on, we're going to be piloting and doing the protocol finalizations for the remaining visits up to age 4, and we have to start developing the protocol beyond that. Again, there's going to be a launch shift up to age 10 for the ages 9 to 10. And annual data releases. This is expected probably after we get through all the enrollment and get all the visit 1s completed. Sometime after that, we'll be starting to release data. So there's going to be publicly available after the data is cleaned and processed and made ready to go. So probably about 2026 or so, I think, when this is probably the first data would likely be released. So, okay, so what does this give us for the department? What can we get out of this? So there's some different things I want to highlight. First one is training and career development opportunities. Second is for manuscript writing. And the third is for opportunities for additional research funding. So let's talk about training and career development first. So possible training areas. So things like pediatric neuroimaging. So learning MRI, EEG, different kinds of child development assessments. Looking at assess child diversity and protective factors in kids. Different measures of family functioning. There's also a lot of community engagement going on in the study. We have a community advisory board to help kind of, I guess, with guide on implemented for a community. I guess I'm not sure how to articulate the best, but we have a community advisory board. Lots of work with bioinformatics. And there's really a lot more. I mean, I think what I'm really trying to press on, this is a really big multidimensional study. There's a lot of, you know, I expect there's going to be a lot of different opportunities for, and there's going to be a lot of different opportunities for training across a lot of different things and beyond what I just listed here. So some training mechanisms I want to highlight that could be use HBCD. One is diversity supplements. I don't know how familiar people are with these, and I'm going to talk about each of these training mechanisms list, but I want to go into a little bit more detail about diversity supplements, because I think this is something people might not be aware of, but I think it could be potentially really helpful. There's also the T32 programs, F Awards, the TRI Career Development Scholars Program, and also K Awards. So let's talk a bit about these. The diversity supplement. So this is funded by NIH. It's for post-baccalaureate all the way up to junior faculty. It's eligible to STEM underrepresented racial and ethnic groups and individuals with disabilities and individuals from rural or disadvantaged backgrounds. This includes low income and first generation college students. So like I would have qualified for these now. So it's up to 2 years support for salary, travel, and supplies. And it's also administratively reviewed. So what this means is, you know, when you submit, say, a grant application to NIH or one of these different kinds of career development awards I'm going to talk in more detail about, it's going to go to a study section first. So that means that it goes to study section, you get different reviewers, they'll review it, and then they're going to meet and they'll discuss it with discuss it in front of other reviewers, and eventually they're going to come up with a score for it, vote on it. The best scoring applications are the ones that move on to the next phase where it's administratively reviewed, and that's can potentially be selected for funding. So this is able to skip that step and go right to the point where it's administratively reviewed. And it's also a lower bar generally than, like, if you're going for a K award, you probably need to have about as strong a bio sketch as you could be writing grants at this point. You just kind of probably just about under, but you need to have a lot of, say, first author publications. You need to really show like you're really developing a lot of expertise in that. These diversity supplements wouldn't require that. So I mean, if you make a case for like somebody needs some additional training or something like that, that this thing could help, you know, you don't have to have as strong a bio sketch. And it's also available to basically all levels. So it can be somebody right out of college all the way up to junior faculty could be eligible for this. You know, and then other mechanisms available. There's the T32. There's the one in our department run by Dr. Kiltz. There's also one in the college public health. These will support pre and postdoctoral trainees, and they also include a didactic coursework, including work on grant development. I'm not going to go a lot of detail on these, but I think this is a great resource, and this, you know, this could also you know, it'd probably be good to hear at a department level more about this too, these programs. Other training support mechanisms, the F31, F32 awards, these are for pre and postdoctoral trainees. So it's like the T32, except the person has to individually apply for these. And they're going to be, again, they're going to be reviewed by NIH study sections. It's supported by different NIH institutes, and let's see. And then there's also the TRI Career Development Scholars Program. This requires people to be a junior faculty member. They're internally reviewed, so that means that they're reviewed by faculty at UAMS and faculty at UAMS. It's supported through the UAMS Clinical and Translational Science Award and institutional funds. And the applications for this are going to be the next round are being due in March of 2023. Then there's also K awards, and this is the most competitive of all the ones I'm talking about here. So they typically support junior faculty. There are some mechanisms for more senior faculty. They've gotten rid of a lot of those. The main ones you're going to see are people basically like the assistant professor level. Usually they're going to target. There's some special mechanisms will target somebody like kind of like an advanced postdoc stage, but for the most part, it's for like the junior faculty it's going to target. Again, it's going to be externally reviewed by NIH study sections, supported by different NIH institutes. So that would be something, again, it'd be good to have kind of more information at department level about, but these are altogether, these are some of the mechanisms potentially available to help support people who want to develop HBCD. Okay, so there's career training, career development for opportunities. There's also manuscript writing opportunities, and I'm going to talk more about what that involves. So again, the first data releases are probably going to be around 2026. This is going to be likely from that remember from the flow chart of visits. This is from that would be from that prenatal visit, and it's going to likely include a lot of things like psychological, social, environmental characteristics of expectant mothers. And the subsequent releases are going to happen after that, which should be happening soon after that. Would be on the neurodevelopment, would include neurodevelopmental outcomes in children that we're following. There'll probably be some biospecimen data, such as drug testing results, available with initial data releases. And the data is going to be publicly available, so anyone should be able to request this data and use it for manuscript development. And I think NIH and I think the scientific community in general is still trying to figure out how to make use of these data sets. But you think about it, like instead, like normally when you get a grant, there's a lot of costs that go towards collecting data, going to collect the data. So the staffing, the participant incentives, and then there's also quite a bit of time it's taking to collect data. What these publicly available data sets are, that it's available right now. So you need much less support to be able to get manuscripts off of these publicly available data sets. And the advantage we have here is we have access to the faculty who are actually involved with developing and conducting the study protocol. So the problem I see a lot of times when, like as a reviewer, either for manuscripts or on study sections, when people try to make use of these publicly available data sets, is a lot of times people don't necessarily really understand the protocol or understand the measures as well as they need to really make the best use of it. So here we actually have the people who can really help navigate people through this and make, and make the best use of that data. Okay, so there's the training and career development, manuscript writing. There's also additional funding opportunities. So the first thing that well, not the first thing, but one of these things that is going to be the renewal of the grant that we have now. That's limited competition. So normally when you apply for an NIH grant, like I said before, it's going to go to a study section. Lots of the applications get reviewed. Generally, about half the applications don't even get discussed in the study section. If they don't get discussed, there's no chance for them to get funded. Of the ones that get discussed, there's much less, a subset of those will be competitive for funding. So it's a hard process to get through an NIH study section to actually get funding. Going forward for us now, we go through a bit different process. So the only people who are able to apply for this are the people who had the funding, had the applications to begin with. And if we're showing good progress and doing well with our first round of funding, nothing's guaranteed, but, and if there's still funding available for the program, we should be highly, highly, highly likely to get this renewed. I mean, so we'd expect this to get renewed. Beyond that, there's also administrative supplements and add-on projects for the HBCD study. So one of the first ones that we've gotten already is a peer navigator supplement. I'm going to talk about this in a minute, this and the other applications in a minute. There's also what we're working on now is a supplement to add more biospecimen collections, actually get biospecimens, delivery biospecimens. We are working with the Arkansas Children's Nutrition Center to do metabolomics assays on from stool samples from the children. I'm going to talk about that in a minute too. And there's also related to HBCD, there's also in the work we've already done as part of phase 1, we are working on new grant applications. So one is adapting a commonly used in-home measure of parent-child interactions for virtual administration. Another one is looking at fetal MRI, especially looking at fetal development with MRI and comparing opiate and non-opiate exposed fetuses. There's also, once the data starts being available, there's going to be data use applications. So let's talk about these. So the Peer Navigator supplement. Jessica's on. I actually kind of stole some of your text from your Blue and You application. I hope you don't mind. I tried to paraphrase a little bit, but I did blatantly steal from it. So, but in okay, thank you. In behavioral health, peer navigators offer their unique lived experience with mental health conditions such as substance use disorders and to provide support folks on advocacy, advocation, mentoring, and motivation. And so for us, what we're using pre-navigators for, so HBCD is provided, gave us additional supplemental funding. So we get pre-navigators on the study and basically help support HBCD participants and make sure they have access to necessary services and resources. So kind of a nice little addition that we got. We weren't expecting that, but within the first year of funding, they made this available so we could take pre-navigators on the project. Delivery biospecimen collection supplement. So again, originally with the HBCD study, they had wanted to get delivery collection of biospecimens at delivery. But this was dropped due to budget constraints. So they didn't get quite as much money as they originally wanted for HBCD. And so this is one of the things they had to cut out. But they've recently requested applications for administrative supplements. So these things that go directly to NIDA, not going through review first, to support delivered biospecimen collection for ongoing studies, HEAL funded studies for 3 years, which happen to be exactly the enrollment period for HBCD. So we're one of 12 sites that are going to be applying for this. The Arkansas Children's Nutrition Center Program Project. For people who are aware of this, the ACNC is a USDA funded research center located at Children's. And they study childhood brain development and especially nutritional aspects of childhood development, things like metabolic processes and impacts of things like child diet and exercise. We're currently seeking, we're trying to get approval from HBCD to do metabolomic assays on stool samples we collect locally to and possibly add in a survey maternal nutrition, which I'm guessing is probably a no-go, but I think we hopefully will be able to do the stool sample assays. And what we're hoping this will do, we're going to do the metabolomic assays on the stool samples collected locally, but the data we get from that, we were hoping would help support an application to do it across the whole cohort. So that's, there's plans right now with the biospecimen collections we're getting. Some things will be analyzed right off, like right away, like we're looking at things like blood tests from the mom, nails from the mom, urine testing from the mom, to look for evidence of drug use in pregnancy. So there's going to be sent off to NIDA, the repository at NIDA, and they're going to be testing those for drug use right away. But some other things that we're just collecting, like stool samples, we don't have plans in place to analyze them yet. So those samples are going to be available for investigators to propose to do analysis with. Okay, virtual home. So the home observation for measurement of the environment, or HOME, it's a widely used assessment of parent-child interactions in home environments. Now, this is normally done in person. So the interviewer would normally go to the family, go to the house, and do the interview there. Part of what started us doing this work is that COVID, so it was gotten to be much more difficult to go in people's houses. And so, but generally though, if we can do this virtually, you could dramatically increase accessibility for this, reduce cost, decrease burden. There's also concerns about going to different neighborhoods and stuff like that. So this potentially could all be done virtually, which we could make a deal for a large multi-site study like HBCD. So during phase 1, we started piloting like the version of the home for the youngest age group, infant toddler. And we are currently developing R1 to adapt and validate virtually administering basically the infant toddler, early childhood, and middle childhood versions of the home. I tried to include a video with this, but I was having a problem with loading, so I'm going to just try and use some pictures trying to explain what I'm doing. So how this works is that we have the interviewer sitting at the desk with a computer, and people usually just use their smartphones, so they can do talking with your family members on a smartphone, and we can do the whole interview like that. So this is just a family. We ask the home asks a lot of different questions about kind of the family environment, the parent, the interaction they have with the kid, and what their activities are like. And the nice thing with the phone, it's kind of like they can turn around, show what the kid's doing, playing, stuff like that. So we really think we can get a lot of what we were originally doing with these in-home visits just with this, the virtual administration. Fetal MRI. So MRI is a well-established and safe tool to evaluate developing, let's say, placenta and brain. Previous MRI studies suggest that both placenta health and fetal brain are affected by maternal opiate use. So as part of the HBC phase 1, we developed methods. Basically, we developed a lot of ways of, so the problem with it, is my wife would always tell me before our children were born that the kids, the babies, the fetuses would move around a lot while as soon as they're laying still. So like, when up and walking around, they tend to be a lot more still, but laying down in MRI, oftentimes the babies are the fetuses are moving quite a bit more. So what we've done during phase 1 is help basically develop motion correction and rapid signal acquisition stuff, so we get fetal brain measures. And we also done some pilot data looking at opiate exposed versus controls, and found that brain sizes seem to be smaller, and there's some other fetal physiological alterations in the fetuses that had the opiate exposures. And we're currently developing an R1 to look at basically look at a couple to do a couple of fetal imaging visits during the pregnancy, to look at both brain and placenta function, and then following it up with a neonatal visit where we give them basically the same HBCD neonatal brain scan protocol, and we also can do pathology on the placenta afterwards, pathology on the placenta, and we're doing some early neurobehavioral assessments on the kids too. So here's just some pretty pictures, but these are fetal brain images from 30 weeks and 6 days old from a study paper we just published. Okay. The last thing I'm going to talk about for grants is the data use application. So based on what they've done there's another study that's ongoing called the Adolescent Brain and Child Development Study, and which is looking at kids from 9 to 10 years old through adolescence, and we're looking at the earlier half of childhood, and we're kind of modeled a similar structure as what ABC is doing, or the Adolescent Brain and Child Cognitive Development Study is doing. What they've done for that study and what we expect to do for this one is that once the data is out there, they're going to start issuing requests for applications, or basically opportunities to apply for grants to actually just use the data. So there's going to be opportunities to look at a range of maternal and child development related domains in this large nationally representative cohort. And again, I expect the advantage that we would have for people who want to use that data is you have access to the faculty who could help you explain how the protocol was set up, how these measures work, and what we're trying to do with it. That's as a reviewer, and I see a lot of these applications on ABCD, and the ones that score well generally have somebody involved with the original project that seem to have a better sense of how to use the measures and how to make the best use of them. So summary. So what's the Healthy Brain and Child Development Study? So it's the largest ever study of childhood brain development. Why is it being done? Part of it's a response to replication crisis. It's also to comprehensively study child brain development and better understand impacts of prenatal and perinatal opiate and other drug exposure. More prenatal, I guess, but yeah. What have we already done for HBCD? So we've done protocol development. We've gotten the SMART IRB approval, site certification, currently piloting the early study protocol. What's next for HBCD? Completing piloting of study protocol, regular enrollment to start in 2023. And what opportunities does HBCD give us? Training, career development, manuscript writing, and more funding. And here is the study website. This is still being developed. It's probably going to be changing as we, especially as we start launching in regular enrollment, but that is where you can also look at what we're doing. Okay. And that's it. Ta-da.
All right. Thank you, Ashley. That's a very thorough and wonderful presentation. Lots of applause. I see hands clapping. We already have some questions. Dr. Dunn, would you like to go first?
Yeah, that's great, Ashley. So you said the first data will be available in 2026. Is that correct?
Approximately. That's not that's just based on what the enrollment window we have and when we should be starting enrollment. So, but yeah, approximately, yeah.
Okay, because I think I love the idea of applying for funding for data. What do you call it? Data use. Or
Yeah, there'll be data use applications. There'll be
So-
requests for data use applications. Yeah, yeah.
how soon would those, so you have to wait for the, is it an RF?
Yeah,
What
the
is?
RFA, the request for application. Yeah, yeah.
So that's not out yet.
It's not out. What people could potentially doing though is for a study that trying to link up with HBCD in some way. So you could try to like somehow
That's,
what. you
yeah,
know, so-
what
yeah.
I'm asking is like, should we be starting to think now? And I think the answer is yes,
Yeah,
about potential data use applications and start planning those so that we're in good shape.
I think so. So, and the challenge, I've already had some meetings with people from public health, but like the challenge is going to be like to get people who can use these kind of data sets, right? So it's not just understanding the measures, but it's also working with these big populations and know how to do the stats in that. But that's where I think the bioinformatics side of things, I think that's where we probably need some help and identifying how we're going to be pursuing those. So, the challenge with these things too is kind of trying to figure out a niche to go for this, right? So I mean, if you just want to do something with opiate exposure on brain development outcomes, that may be harder. You have to somehow, and it's not like there can only be one application doing that, but you have to kind of, you have to have some type of angle with it, right? So I mean, it's going to be, and that's kind of give me the challenge to kind of figure out how to use this data, because it is publicly available. Anyone can apply for this. How do you set yourself apart too?
Yeah, well, and I'll say, I mean, I think that Ashley, you are very well suited to help us figure that out.
Well, hopefully.
All right, great. That's great.
All right, we have time for more questions, and I may not be able to see your hands if you're raising, so either text or just start talking. I'll lead off with a question. You mentioned that the HEAL is specifically focused on neonatal exposure to opioid misuse, but it seems like in the criteria you listed, it'd be much easier for subjects, participants to meet the criteria for alcohol,
Yeah.
nicotine, or marijuana use. So is there going to be some like minimum percentage, like at least 10% or 15% must meet opioid misuse? Is there been discussions about that?
There's no, so first, no, there's no requirement for opioid misuse. So that what they're, but they will have to, so 12% of the sample will have to meet those opioid use criteria.
Okay.
And what that means, what they're trying to really do is get a range though, right? So they're trying to kind of over sample a bit for heavier substance use than the other ones, but they're trying to anyone who doesn't meet that threshold would be, still be in the sample. And that's just how we're trying to target. But like when this data is all available, people can look at this data how they want. So it may be that some people, other people would define opiate exposure in a different way than we do, or we're doing for enrollment. But yeah, so yeah, when we originally planned the study, we thought we were going to have 25% of sample with opioid use disorders, and then what's what the protocol developed and what NIH wants is that no, it's a more lighter threshold that we're going to be going for.
Excellent. Other questions? I have another follow-up if not. So how is the HBCD handling comorbidity, like opioid use with comorbid alcohol or nicotine use?
We're recording it, so there's no one thing I didn't mention, that there's really minimal exclusions for the study. So like we are trying to basically be as representative as possible, and with the hope that with the large sample size we could look at different factors or control or covariate for things. But yeah, they're not trying to get like a only X substance use groups at all. It's just what we get who meet the criteria, and they could meet criteria for different things.
Yeah, that's probably more naturalistic of what you'll expect.
Yeah, which is what they're going for. What we're going for. Yeah.
Excellent. Additional questions?
Dr. Acheson, this is Srini. Great presentation. Just really interested about, are we looking at the stimulant use in this? I know you mentioned about alcohol, opioid, marijuana.
So as of right now, that is not one of the criteria that one of the groups that they gave. And I'm trying to remember what the rationale, there was pushback. As you imagine, like we have lots of lively discussions in these work groups over the past, since, for the past couple of years. And I'm trying to remember the rationale for not targeting substances. I think the idea was that they wouldn't get somebody's exclusively stimulant, that they would likely be captured with one of these other things, so I think that was like you could get if somebody with has, yeah, I think it was just that they didn't feel like they would get a lot of only stimulant user, that we'd likely capture them through one of those other groups too.
But you know, for the opioid use, the threshold is only 2 weeks of prescription opioids, right? For somebody who actually used prescribed stimulants like, you know, Adderall and other stimulants prior to pregnancy or just close to the pregnancy, will that also affect the data that you're collecting if they're also?
We aren't specifically targeting those people, but they wouldn't be excluded. So I mean, it's like, you know, and I expect we'd probably see some of that, right? I mean, so I think, you know, like a lot of the substance exposures generally, people could easily meet those, like a lot, and I think people want people would just meet them, but like see some of these things too. But you know, people could pretty easily meet these just during, you know, they didn't know they're pregnant, you know, and they, you know, just did
Right.
normal behaviors and stuff. So, you know, but yeah.
Yeah, it's very interesting. Thank you.
Thank you. Thank you.
Dr. Acheson. Great
Hi.
presentation.
Thank you.
I just wanted to ask you, I noticed that we didn't address the issue about patients with mental health disorders.
Yeah. There's not exclusion for it. So like you know, that's one of the things, you know, so when we're thinking of early on, we're thinking about this and we're thinking of targeting patients with substance use disorders, or that we're thinking we had to target patients with substance use disorders. You know, substance use disorders usually don't happen by themselves, right? They tend to go along with other things. And so that's why we thought that we would need to actually target people with other things without the substance use disorders. That's not how they're having a substitute matching, but we're also not excluding people with mental disorders. So we, you know, there's no exclusion for having, like, you know, being treated on antidepressants. There's no, you know, like that, you know, so people could have a variety of different disorders and still be, there's really minimal exclusions for the study.
So I guess when it comes to the percentage of how they're going to recruit, where are they going to fall into that category? Will they fall into that 25% or will they fall into that 75%?
It depends if they meet whether they meet substance use or not. So like if they don't meet the substance use thresholds, they'd be in the 75%. If they do, they'd be in the 25%.
Okay, thanks for that clarification.
Oh, no problem. No problem.
We're at the end. We might be able to squeeze in one more question. I don't see any takers, so please join me in thanking our speaker. Thanks, Ashley.
Thanks, everybody. Thank you.
Go science.
Go science. Yay. That's how I end all the meetings. Thanks, everybody.